This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 1191, recorded on January 31st, 2025. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from Western Massachusetts, Alan Dove.
Good to be here. And let's see, it is kind of crummy weather here. We've got light rain. It's 41 Fahrenheit, which is the warmest it's been in quite a while. But it's wet and cold and foggy and supposed to stay like that. At least it's not snowing. And then I think we're getting another deep freeze coming up.
Yeah, here it's 7C. It's raining, and it looks like, it's going to go back into the deep freeze. So I guess that through February.
Yeah.
It's cloudy outside, but at least it's not a deep freeze right now.
Right.
Okay. If you like these programs, it's just Alan and I.
Yeah, it's just us. It's just, that pause there was like he was getting ready to welcome in the other guests, and I was thinking, yep, he's going to remember there aren't any.
No, I was just going to do something else, but I wanted to tell you that Alan and I have done this before.
Yes, we have.
The very beginning. So I started this with Dixon, and then it became clear that Dixon didn't want to be stuck every Friday doing this. So Alan joined us, and then Dixon took off. He said, I'm going to India for a month. And so Alan and I did a bunch together. So, yeah, and then recently we've done some as well. Well, that's why we have a big stable, right? So we can
swap people in and out. We've even done a couple of episodes without you. We're not terrible, but we like to have you.
If you like these episodes, these science programs. Now, if you're interested in science and you want to know what's going on, we are something you should consider because the flow of science information is getting impacted, to use the word.
Yes.
So listen to us, and we do this on your donations. We don't do ads or anything. We don't take money from anyone except you. So please contribute. Microbe.tv/contribute. ASV 2025 will be held in Montreal at the Palais des Congrès de Montreal, July 14th through 17th this year, 2025. The website is asv.org/ASV 2025. And it's too late for the travel awards.
If you're listening to this now, you already missed the deadline for a travel award.
Yeah. And as far as news goes, well, everyone knows there's a lot of news swirling around these days. We have two articles about the freeze, the Trump freeze on meetings, science meetings, travel, communications, hiring. This is all having to do with HHS. So it includes NIH, CDC, FDA, so that you can't communicate, you can't have meetings, the budget is frozen. And so the excuse is so that they can determine what it is you're going to hear. It sounds ominous, doesn't it?
Yes.
They want to censor it and say, no, you're not going to hear this. You're not going to hear about the goodness of HPV vaccine. You're not going to hear. That's what I'm afraid of, frankly. I mean, but that's so we have a science article called "The Trump Hits NIH with Devastating Freezes on Meetings, Travel, Communications, and Hiring." So everybody's freaking out. This is by Jocelyn Kaiser.
Yep.
Everyone's freaking out. When you disrupt things like this, that has long-term effects. It's hard to get back to normal right away. NIH in particular, I mean, it also affects the other HHS agencies. You can't hire people. You can't have study section meetings. Study sections are made to decide who gets money, research funds. And you can't, there's no new funds dispersed.
They can't hold those. They can't go to or organize or host conferences. Basically the whole operation of HHS is jammed right now. And when Vincent said impacted, I'm not sure whether you meant impact as in effect or impacted like a colon, but I was thinking like a colon.
I was thinking like a colon, yeah.
Yeah. Yeah, so the whole system is just jammed. And this is government dysfunction. This is chaos. This is what people voted for. So here y'all go.
Yeah, I don't know that most people have any idea what's going on because most people don't pay attention to the news, right?
I think it's pretty hard to avoid, but people will notice when it starts having an impact on their day-to-day lives. Sadly.
And we have another science article entitled, "Is Your Research on Trump's Spending Hit List?" How the White House's pause in federal spending could affect academic science. I don't know who this is by. Jeffrey Mervis. So this is also about the spending freeze, and the idea being that when it starts up again, it may not be everything. There may be some cuts to things that they don't like. So this is bad. It's really
for
bad.
a computer analogy, I know we have a lot of IT folks in the audience. It's like if a new user on a Linux system found a list of running processes and just went through killing any that they didn't recognize.
Yeah.
Imagine what impact that would have.
Yeah, that's good. I like that. So if I were still in the NIH game, boy, I'd be freaking out.
Oh, yeah.
So I have I have sympathy for our colleagues who are stuck in this. In fact, we have a pick that relates to it, a way that you can contact your congressperson about this coming up, because you should write and complain, even though everything's on a partisan line now. Anything happened with RFK Jr.? Did they vote?
They were, I am not really if the news has like a Trump headline in it, I don't, because I'm not going to visit that. But last headlines I saw, they were still grilling him, so I don't know.
Let's see.
I mean, if he doesn't make it, then some other idiot who's probably just as bad is going to get put up in his place, because that's the whole point, is to wreck the system.
Yeah, they haven't voted yet.
Yeah.
The Hill says his nomination is in trouble.
I hope so.
The thing is that the numbers go like this: party line vote, and then
there are like 3 people who might.
Murkowski.
Yeah.
Who's the other? Collins.
Right.
And in this case, McConnell doesn't want to vote for him. That makes it 50-50, and then the schmuck casts the deciding vote.
Yeah.
And I mean schmuck. I've just never seen such a schmuck in my life. There's some poll office said there's one doc in Congress who might vote against RFK. I have not been watching the hearings, but people have been telling me, and he's just totally out of his league, and the Democrats are ripping
he's
it.
completely unqualified for any government post, let alone this one.
Bernie Sanders brought out this onesie. It's some kind of a thing that you wear, like a baby would wear.
Yeah,
right?
yeah.
It says basically, "Don't vaccinate me," sold by his company. And Sanders said, do you endorse this? And he said, I don't have anything to do with that company anymore. And just lie.
Yeah.
He also would not agree not to take litigation money. Like his company is suing Merck over the HPV vaccine. He'll still take it. He said, no, I'm not going to agree not to take. I mean, in the old days, that was a killer,
Yep.
but no more. So by the next time we record, I'm sure this will be resolved, and hopefully
Probably in some horrible way.
get your kids vaccinated.
Yes.
Do it as soon as you can, because who knows what's going to happen to vaccine access anymore. It's a very sad situation that, science has, I understand that the science in part is political, but it's never been to the point where you can just lie about science and get away with it.
Yeah.
They would say, "Oh, did you say this?" "No, I never said that." And it's in the record that he said. I don't know how you can say that. That used to be the end of your career. Now it's not anymore.
No, it's not. Politics is completely unmoored from reality now.
Do you think it will ever get back?
I don't know.
Wow. Well, then let's talk about science, because that's more interesting.
Because we still can, yes.
We have two papers for you today.
Yeah. So
give us a summary, Alan.
All right. So I can do the
Bats.
Initial bit and the summary. First one is from Science Translational Medicine. It's prior vaccination prevents overactive.
You want to do that? Let's do the bat one first.
Oh, do the bat one. Oh, sorry. That's right. Yes. I'm on the wrong one.
Although I think.
Wait, I thought we were doing. No, I thought this was the snippet. The prior vaccination is the snippet.
It doesn't really matter. You can do that first.
We're going to end up talking for a while about both. You want to do the vaccination one first? That's how I had them.
Yeah, it's fine.
Okay, so prior vaccination prevents overactivation of innate immune responses during COVID-19 breakthrough infection. The first author is Leslie Chan. Senior author is Catherine Blish. And the team is from Stanford University School of Medicine in Stanford, California, the Harvard T.H. Chan School of Public Health in Boston, Stanford School of Medicine, and the Chan Zuckerberg Biohub. And I noticed that Leslie Chan is neither at the Harvard T.H. Chan School of Public Health nor the Chan Zuckerberg Biohub, so I think she's a different Chan. And let's just get to the right thing. There we go. So a short summary. In severe COVID-19, the overactivity in the innate immune system causes a lot of the damage. We know that. And these folks looked at the effect of prior vaccination on innate immune responses in vaccinated people who got so-called breakthrough infections, a term neither of us likes. They found major differences in gene transcription and translation patterns, cell interactions, and patterns of which cells respond between comparing initial pre-vaccination infections with these post-vaccination infections. In particular, monocytes and natural killer cells seem to be less activated in the post-vaccination, the breakthrough infections. Interestingly, women had higher expression levels of inflammatory markers in these breakthrough infections, while men had more inflammatory markers in primary infections. The importance of this study is that it shows prior exposure to SARS-CoV-2 vaccine significantly reshapes not only the adaptive immune response, but also the innate immune response to a subsequent infection, which may explain some of the reasons vaccines protect from severe disease. So it's not just about building antibody and T-cell responses.
So let me explain why I don't like breakthrough infection. So, I mean, the key to this paper, it ends up that the paper is very interesting, but not for the reasons they state. So they say at this stage, most infection in the COVID pandemic, most infections are breakthrough that happen in people with prior immunity because everybody's been vaccinated or infected or both, right? The implication is that they shouldn't be happening, right? That the vaccination or previous infection should be preventing these. And so they say we have to look at the immunology to see what's going on here. But you will never behind a vaccine that prevents infection. So it's wrong to look at this as an anomaly, okay, these infections.
Right.
Fortunately,
They're not breaking through in the sense of, a security breach. There was some sort of absolute barrier and these got through. This is a normal thing that happens when people have been vaccinated. You can still get infected. Sometimes if you just, you got vaccinated, your antibody levels are high enough, the infection may not go anywhere. You may not ever be able to detect it. That's what's referred to sometimes as sterilizing immunity. But that's kind of the exception rather than the rule. Most of the time, you got vaccinated against poliovirus as a kid and you get exposed to polio that's circulating in the environment. It gets in, it starts to replicate and your immune system shuts it down. You got infected, but it didn't go anywhere and it didn't cause disease. So the idea of a breakthrough infection is not something that we really...
So I guess they were motivated to understand the immunology of breakthrough, which I would have said is not a project, but they end up finding things that are interesting.
Yeah. Yeah, because I mean, as I said at the top, we do know that there's in severe COVID-19, which can happen even if you're vaccinated, if you have comorbidities, if you've got immune system problems, then even if you've been vaccinated and maybe even had prior infection, you could still get a severe case of COVID-19. And so my take on this is, yeah, we should study this not because we're trying to make the perfect vaccine, but because we want to know what causes severe COVID.
Exactly. So essentially what they did was to look at vaccinated and unvaccinated people during the Delta wave, April through December 2021. I can't even remember that era. So that's a time when there's still a lot of people not yet vaccinated. And none of these people had severe disease. They're all moderate disease. And they do single-cell RNA sequencing, all right, which basically said you got to get a single cell, and then you extract the nucleic acid from it, and you sequence the RNA. And that tells you what mRNAs are present. Okay?
In that particular cell.
In that particular cell, you can compare it between unvaccinated. So there are three populations. There's healthy controls, right? They don't have any COVID. Then there's the and then there are people with COVID. There's the unvaccinated and the previously vaccinated, right?
And with this single cell sequencing, we should note for people who are new to this, you don't just sequence a single cell. You sequence a bunch of single cells, but the important thing is you can drill down to the data to the single cell level.
Yeah, I think you use flow cytometry to get lots of single cells, right? So it's a machine that puts single cells through a gate, and you can take lots and lots of single cells based on different criteria, cell surface markers, cell size, and so forth. All right, and so then they take they have single cell RNA sequencing, mass cytometry, and plasma proteomics. So a lot of this big data kind of science, and then they crunch it to get some conclusions. Okay, so in the beginning we have. By the way, I was going to say, whether a paper is a snippet or a paper is often moot.
It's often arbitrary, yes.
Because I'll see papers that look interesting, and it's hard to know how long the discussion will go or even how long the paper. So sometimes they end up being different, but it doesn't really matter. The point is we discuss them.
We're discussing two papers, yes.
All right, so I have healthy controls, HCs, the unvaccinated infected, that's UVI. Unvaccinated infected. That's pretty straightforward. Unvaccinated infected, UVI. Yes. And then vaccinated and infected.
Which is VI.
VI. Which is interesting that they don't put breakthrough in the abbreviation.
Right.
it would be VBI, but they choose just to do VI. It's interesting. Cool. Let's see. Healthy controls, uninfected at the time of sample collection. That's 30 weeks after their second dose. So the healthy controls are vaccinated as well. The vaccinated ones got 2 doses of Pfizer or Moderna. One person got a Janssen. Remember Janssen?
Oh, yeah. Janssen, Johnson, and Johnson.
And then they draw blood samples at different times. And they all had mild to moderate infection. No significant differences in the days since symptom onset on sample collection. Okay, how does vaccination alter the response to SARS-CoV-2 infection? Again, single cell differential expression in these 3 patient groups. They have all kinds of programs nowadays that you can just apply to your data and, packages. One of them is called a blood transcriptional module. So they're all the transcription that happens in blood. Compared to healthy control samples from both infected groups, upregulated genes enriched in viral sensing, type 1 interferon response in multiple cell types, including B cells, monocytes, T cells, and NK cells. Not surprising.
That all makes sense.
It will make sense. So prior vaccination affects both innate and adaptive. Actually, so that's both innate and adaptive immunity. So the prior vaccination impacts both. Now, they know from previous work that these these infections are associated with a greater proportion of monocytes compared to primary infections for whatever reason. So they said, let's look at the monocyte compartment. We can purify monocytes, and they have classical CD14 positive and non-classical CD16 positive. The same in all the groups. No differences there. However, when comparing the CD14 positive monocytes from the UVI and VI, the CD14 from UVI's upregulated genes enriched in, so UVI are the unvaccinated, infected, upregulated genes that are enriched in type 1 interferon, innate antiviral response, and also had a greater pro-inflammatory response. And consistent with that, these monocytes upregulated an interferon-stimulated gene pattern. So greater interferon-related induction, which is consistent with higher inflammation. Okay?
Yeah, so the innate immune cells in the unvaccinated people who are first encountering this virus, those seem to be more active than in people whose immune systems have previously seen it.
Lots of figures here.
Lots of figures and complex figures.
Take a long time to load on my not very old computer. Okay. Figures break up the text, so I highlight stuff and then I can't remember.
Oh, yeah.
The beginning of the highlight. Okay. So I wanted to just say that. So what they find so far is that vaccination attenuates inflammatory profiles in both classical and non-classical monocytes in the VI vaccinated infected participants. Attenuates, which means it reduces them compared to the unvaccinated, right?
Right.
So that's interesting that there is an attenuation of an inflammatory profile.
Yeah, so because the innate immune cells, I mean, they're not supposed to, like the way we think of them classically is the vaccine is triggering the antibody response from the B cells and the T cells, and that's our adaptive immunity, and so the vaccine should be all about adaptive immunity. And yet, it turns out if you got the vaccine, then you later get infected, it is changing the innate immunity pattern. So something's different about the whole response.
Yeah. And these patients don't differ in the kind of disease they have.
Right.
So it's curious. I mean, it's a very interesting observation, but I don't know what it means because these patient groups are about the same.
Yeah.
All right. So then they take monocytes in ex vivo, right? So they're going to do some experiments outside of the person from which they got them. And they both respond to stimulation, but the unstimulated monocytes in the UVI participants had elevated interferon gamma inducible protein 10 compared with unstimulated monocytes in VI participants. So this is an interferon gamma inducible protein, again, involved in some kind of inflammatory process. Monocytes from VI participants had reduced protein expression of CD123. So what does that mean? And some other proteins as well. The reduced protein expression of IP10 and CD123 is consistent with a reduced inflammatory monocyte response in breakthrough infections compared with primary infection. So essentially, they've put these cells in culture and done some things to them, stimulated them and measured it, and they get a similar response to what they've already found with the single-cell RNA-seq, in that they have a reduced inflammatory response in these monocytes in breakthrough compared with primary. So again, not that this has anything to do with breakthrough, but it has something to do with being immunologically experienced for this pathogen, which can be useful. They look in the plasma. I like Olink's inflammation panel. Okay. A plasma proteomic assay. Look at all the proteins in the plasma. And plasma from both participants, UVI and VI, had elevated concentrations of a bunch of inflammatory proteins. And I don't have to name them for you, compared with plasmas from healthy controls.
Makes sense.
In the UVI participants, certain plasma protein concentrations were significantly elevated compared with VI. But in contrast, matrix metalloprotease 10 was elevated in plasma from VI, okay, compared with the other two groups. And this protein, MMP10, has a protective role, at least in other infections. It can cause some immunosuppression, so you don't have immunopathological damage. So remember, that's the that's the VI participants. The vaccinated, infected, with less of an inflammatory response. So the matrix metalloproteinase elevation is consistent with that. So some things are down, like your inflammatory proteins are down, but the ones that promote anti-inflammatory are up. Makes sense.
Yep.
They do some experiments to explore communication pathways that are driving the activation of monocytes in these infections. They use a package called multi-niche-net to look at receptor-ligand interactions and signaling and CD4, CD8 cells, monocyte. I don't quite get what they're doing, frankly, but I will give you the summary. It's all in software.
Yeah.
A breakthrough infection in VI results in a less activated transcriptomic profile in monocytes than primary infection in UVI participants. So this data that they just generated on interactions is all consistent with that.
Yeah. Yeah, you have a different set of interactions depending on whether you were vaccinated or unvaccinated when you got infected.
Okay. NK cells. Then they're going to look at another kind of cell. Because remember, we're looking at so far monocytes of two kinds, classical and non-classical. Hopefully you got that so far.
Yep.
NK cells, I love when they look at a marker and they say C56, a cell surface marker, dim, and C56, bright. Okay. And again, they look through lots of proteins in these NK cells, and I'm not going to go through and tell you them, but essentially what they find supports an active role of NK cells in SARS-CoV-2 infection with a greater inflammatory and activated response in unvaccinated individuals compared with vaccinated. All right, so essentially you get greater activation of NK cells in the unvaccinated cohort. Infected and vaccinated. Unvaccinated and infected. So not only monocytes, but macrophages as well. I'm tempted to tell you some of these proteins, but it doesn't really matter. It's just, again, it's a set of proteins.
And kind of once you open that can, where do you stop on the list?
There's a lot, but
there's a lot. Yeah.
You need to explain what they're doing.
Right.
But they're enriched in these patients that have no vaccination.
Yeah. And the ones, at least the ones I recognized, are kind of associated with the inflammatory, pathogenic, cytokine storm sort of bad immune behavior.
Yeah, so NK cells are involved in pathogen clearance, and they can cause damage.
Yeah.
Yeah, so that's reflected in the findings of they're more highly activated in unvaccinated individuals. They also did this signaling analysis with NK cells that they had previously done with monocytes, they can try and infer what kind of cells NK cells get signals from, and they conclude that NK cells from unvaccinated infected participants receive ligand receptor interactions that promote cell proliferation and chemoattractant secretion, whereas NK cells from vaccinated infected get ligand receptor interactions that limit NK cell proliferation. So if the NK cells proliferate, they're going to make more proteins that cause more damage, and secreting chemoattractants will bring other kinds of cells into the infected area, potentially causing damage. Right. So this is consistent with the dampening of these responses in vaccinated infected individuals.
Right. So in the vaccinated group, they're kind of, pulling back on the chain on the NK cells and reeling them in.
So just like monocytes, NK cell responses differ depending on whether you've been vaccinated. All right. Next section. Innate responses are predicted to differentially regulate adaptive responses. We all know that innate responses can regulate adaptive responses, and they wanted to see differences in adaptive responses, because so far we've been focusing on innate responses, right? And so they want to see differences in adaptive.
This is a two-way street. Innate and adaptive immunity talk to each other, and each can regulate the other.
What language do they use?
They use one that we do not know.
Who is it that said...
We're not getting Duolingo for immunology anytime soon.
Oh, yes. Josh Lederberg said, bacteria are always talking to each other. We just don't know the language.
Yeah.
All right, differences in adaptive response. No differences in the frequencies of naive B cells memory B cells, intermediate B cells, and plasmablasts, or neutralization activity of plasma against SARS-CoV-2 Delta when you compare UVI and VI. Okay, so at that gross level, no differences. Transcriptomic level, they have some indication of an inflammatory role of certain B cell subsets. In a T cell compartment, again, no differences in the frequencies. We just went through the different kinds of B cells, right? So in the T cell compartment, no difference in frequencies of CD4, CD8 kinds of cells. But they do see a transcriptional difference in a specific T cell compartment, which is called the T central memory. So there are all kinds of T cells floating around, and T central memory are an important one. It was as opposed to effector memory. I just don't remember which one's which. I think effector memory is residing in the tissue where the infection first happened.
Right.
And central memories are circulating.
Right.
That could be wrong.
Yeah. This is why we need Brienne.
Yeah, I would just ask Brienne. Anyway, there were transcriptional differences in certain kinds of T memory cells. And so I can summarize that as prior vaccination gives you reduced transcriptomic activation in naive B cell, intermediate B cell, memory B cell, CD4 T, central memory, and CD8 T effector memory subsets, and breakthrough compared with primary infection. So there are some differences in breakthrough versus primary, even in the adaptive arm, according to these transcriptional analyses. I don't need to tell you what they are. You just need to take the word that there are differences, which are echoing the differences that we've already looked at in the innate immune responses.
Right.
Now, we know that COVID-19 outcomes differ in males and females. So they wanted to see if any of these things that they find that are different also differ by sex, right?
Right. So far we've been talking about the averages for the whole population of healthy controls, unvaccinated and vaccinated. Now they're going to break it down by sex.
Right. So sex stratified analysis revealed a female-specific upregulation of genes enriched in viral sensing, type 1 interferon, antiviral response, and activation in CD14 and CD16 monocytes. All these things we've been talking about in VI versus UVI participants.
Right. So
all
women have more activation of these genes
right.
if they've been vaccinated compared to unvaccinated.
Whereas in males, the only things upregulated are in the samples from the UVI participants.
So it's the other way around.
Yeah. So you unvaccinated, infected.
This is likely related to the differences we saw in the pandemic with severity and mortality rates.
Yeah.
Where men were at much higher risk of getting severe COVID.
So they look in the plasma in the NK cell compartment of males versus females, right? It's same result. The kinds of activation differences we already found is accentuated in females. More activated, the NKs are more activated in breakthroughs than in males. In the plasma, no differences in the cytokine concentrations between male and female, unvaccinated, infected, but samples from vaccinated infection, females had an elevated expression of a variety of markers involved in what can I say? Longevity, abundance of mature dendritic cells, cytokines, and so forth. So basically, the SARS-CoV-2 infection gives you a better innate immune response in vaccinated females than in males. This is not surprising because, and this is evolutionarily preserved. We see it in other species because the female is the reproductive part of the species, and they have to be protected. So that's the logic for these kinds of observations. The differences, by the way, go away at older age.
And if you're interested in more on that topic, Sabra Klein, right?
Yeah.
We had her on. Very, very interesting.
A long time ago.
Yeah.
She was going to come back, and then something happened.
Oh,
maybe it was the pandemic. I don't remember.
I thought we had her on during that, or maybe. Well, I lose track of
she was
that
going to have. She was going to be on more recently.
2020 and surrounding years is kind of a blur.
And then finally, no, not finally, but another experiment comparing, again, these kinds of interaction predictions between innate, in this case, innate immune cells and adaptive immune cells between males and females. They can identify interactions from monocytes and NK cells upregulated in samples from male vaccinated infected participants. And these interactions suggest that CD8 positive T cells in breakthroughs are more activated in male participants than in female. So more activated, which would mean more inflammation. And because monocytes and NK from female VI were more activated during breakthrough infection than those in males, it suggests that these innate immune cells may drive greater immunosuppression, resulting in greater innate immune activity in females than males with breakthrough infection. So this is a little bit of a mixed bag here because they want to figure out why women are better protected, right? And so it does track with the VI versus UVI. If you're vaccinated, you have a less inflammatory profile.
But if you're male and vaccinated, it's not. So it's not completely the reverse in men and women. So everybody gets this consistent response where you get less inflammatory response on reinfection or on post-vaccination infection. But if you're female, you don't get as much of a lessening.
Yeah. It's a less. If you look at the bottom of page 11, left column, the last paragraph, because monocytes and NK cells from female VI participants were more activated during breakthrough infections than those in males. Okay. So that's weird because we're arguing that VI are suppressed, right?
Right.
But not in women, not in females. Okay. They're more activated during breakthrough than males. This suggests that these innate immune cells may drive greater immunosuppression. And then the sentence breaks down.
Data suggest, right. Yeah.
I don't know what they're talking about.
Something happened in the edit there.
Sex differences result in greater innate immune activity than males. So basically, that's what we're seeing. Females have greater innate immune activity, even though the overall idea in this paper is that if you're vaccinated, you're kind of suppressing the innate immune activity, right? And even the adaptive.
But it's not
activity.
As suppressed in women as it is in men. Or it's
That's right.
I think that's
right. Yeah. So maybe there's the female need for a good immune defense that overrides that, right? I don't know. All right. So then they said, does this apply to other COVID vaccines?
This read like a Reviewer 3 section.
Yeah.
Right. So they've looked at the Pfizer and Moderna vaccines, and then for whatever reason, they decided to look at people from a UK cohort. The UK cohort had received the ChAdOx1 vaccine, which is different.
It's an adenovirus vectored, right? The chimpanzee adenovirus. And so apparently they published their data, right? Their RNA-seq data.
Yep.
So this group could get it. But they don't have a lot of participants.
Yeah. 16 study participants. So it's a small group. They didn't really have the statistical power to look at the sex differences. But what they were able to look at seemed consistent with what they saw in there.
Yeah. So both monocytes and NK cells, vaccination attenuates inflammatory responses. So that's the key finding here. If you are vaccinated, you attenuate your inflammatory response. So that's very interesting, right?
Yeah.
Because as Alan said at the outset, inflammation can be the basis for pathology. So we want to know how to regulate it. And I think it was very funny in the abstract. They say, these results underscore the potential of harnessing vaccines in mitigating pathologic immune responses. Well, isn't that what vaccines do?
Well, yeah, but it's not conventionally the way we think of vaccines working.
Yeah, we don't think of that, responses.
We think of them as activating the adaptive immune system, which is then more active and more specific when you encounter the infection and therefore takes it out. But it turns out, probably not surprisingly, since it's the immune system, that it's more complicated than that.
All right, so what you're saying is that we have to figure out now the mechanism by which vaccination dampens inflammatory responses, and then see if we can design vaccines that do that really well.
Yeah. And maybe even come up with interventions that could do this, post hoc. So somebody gets a viral disease and seems to be headed down the wrong road with it, where they've got the cytokine storm and the immune activation, and we know this is going on in COVID-19, but it happens in a lot of other conditions too. So I'm thinking, somebody ends up in the hospital with a bad case of flu. Very similar situation. You may be too late to treat them with antivirals, but their immune system is out of control. This provides hints for where to look for mechanisms you could maybe step on to deal with that.
They say in their discussion, the ability to clear infection without driving marked innate immune activation may be the key to preventing severe disease. It's always been thought that the individuals who, when infected with, say, SARS-CoV-2, when they mount a strong interferon response, it really knocks down virus levels. Then the interferon response is controlled. They do the best. Whereas if you have a poor interferon response, then viral replication goes out of control and the interferon response tries to catch up by amplifying itself, and that's bad.
Yeah.
Right. That's the simplistic explanation of what's going on here. Now, if only we knew how things worked, then we can start messing with them, but we don't. Anyway, I thought that was cool.
Yeah.
Just, folks, no breakthrough.
No, don't use breakthrough.
I said prior vaccination prevents overactivation of innate immune response during a COVID-19 infection.
Yeah.
Prior vaccination, you got it all in the title, prior vaccination. You don't need breakthrough.
Yeah. Breakthrough should only apply to your science, not infections.
Breakthrough prize. Isn't there such a thing?
There's a breakthrough prize, yes.
What do you get that for?
I don't remember what that one's for. I'm not in the running.
It's okay. All right, we have another paper for you.
Yes,
Tell
we
us
do.
about this one, Alan.
All right, so this one First of all, the authors, the co-first authors are Ariadna Morales and Yu Dong, and the senior author is Michael Hiller. And the group is from the Lowe Center for Translational Biodiversity Genomics in Frankfurt, the Senckenberg Research Institute in Franklin, Goethe University in Frankfurt, Goethe University of Frankfurt, the University of Edinburgh in Edinburgh, UK, Zhejiang University in Haining, Max Planck Institute of Molecular Cell Biology and Genetics in Dresden, Dresden Concept Genome Center in Dresden, University of Saskatchewan, Saskatoon, Canada, University College Dublin, Ireland, the Max Planck Institute for Psycholinguistics in Nijmegen, the Netherlands, SUNY Stony Brook, New York, Tel Aviv University in Israel, University of St. Andrews, UK, Institut Universitaire de France, Paris, University of Montpellier in Montpellier, France, Queen Mary University of London, Robert Koch Institute in Berlin, Texas Tech University in Lubbock, Royal Ontario Museum in Toronto, the University of Waterloo in Waterloo, Ontario, and the University of British Columbia in Vancouver. Got all that?
It's huge.
A very, very international group. So, the title of the paper is Bat Genomes Illuminate Adaptations to Viral Tolerance and Disease Resistance. And as a short summary, we've talked about this a lot on TWiV. Bats are major reservoirs for viruses that can spill over into humans and domestic animals. But how do the bats survive all those infections? So this enormous team did a massive analysis of 115 mammalian genomes, including bats and, for comparison, rodents, and looked for immune system genes that appeared to have undergone major selection in bats. The results show a lot more immune system selection in bats and their immediate ancestors than in other mammals. Zooming in, they looked in detail at a gene called ISG15, which is upregulated in COVID-19 and believed to be involved in disease severity. ISG15 from bat species commonly associated with coronaviruses significantly reduces SARS-CoV-2 production in cells, but the effect differs between bat species. And the importance of this study is that it reveals a rapidly evolving antiviral gene and also uncovers a long list of other potentially important immune adaptations that could contribute to bats' powerful antiviral defenses.
All right. By the way, the Breakthrough Prize
Yes, they should get the Breakthrough Prize.
is established by Sergey Brin, Priscilla Chan, Mark Zuckerberg, right. Blah, 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 right? So, and they fund it with their personal fortunes.
Right.
There's one in math, there's one in physics, and there's one in life sciences. I don't know. So I think it's one of these over publicized things. All right. So this is quite interesting because there's a lot of bioinformatics here, but there's also some experiments, wet experiments, I should say, which is unusual because usually you get a paper full of genomics, and that's fine. But here, I like when they do experiments that are suggested by their data, right?
Well, and when you have a group this big at this many different institutions, you probably have access to a lot of different techniques.
Yeah, I think that's why the group's so big is that they decided at some point to do some wet experiments and needed to recruit because the people sitting at keyboards, I don't know how to do any of this. So
and the people pipetting are like, I don't know about that algorithm.
Yeah. All right, so everybody knows the bat shtick. Bats, are the only mammals that can fly. 20% of mammal species are bats. Now, mice can't fly, but they're 40% of mammalian species. Bats harbor lots of viruses, and they can infect other species, as you know, and have been the source of many zoonotic infections. Viruses from 31 families have been found in bats. They have a lot of coronaviruses in them, among other things. They make the statement here that infections in bats are largely asymptomatic. I object to that statement. Have you looked at every infection in every bat everywhere?
And the ones that are symptomatic, you probably don't find those bats.
No, right. They're in the back of the cave dead.
But yeah, what they mean is viruses that are severe in humans are often, you find bats that are carrying virus, and you can get, live virus out of the bat, and the bat seems fine.
Yes. And
So
they've
they
done a
do have
few
this. They do have this phenomenon where they can carry lots of these viruses that we consider serious without really being hurt.
I mean, we've covered some papers where flying foxes are carrying Nipah virus, right?
Yeah.
They seem to be okay. And in fact, there are some experiments where people infect, you'll see in this paper, with viruses that make humans sick, and the bats seem to be okay. So I agree, but to say they're largely, I don't think that's correct. I think that's too broad.
Yeah.
They may be. Here it is. Experimental infection with Marburg or SARS-like viruses, productive viral infection, but no clinical signs of disease in bats. So how can this be? Well, as we just said in the previous paper, inflammation is a big part of tissue damage, and it's thought that bats can limit that. I remember many years ago, first bat genome sequence showed amplification of genes that are involved in mitigating oxidative damage, because when bats fly, they generate lots of oxygen radicals, and those have to be handled, right, by the immune system. And so the mechanisms can probably translate over to virus infections, limiting inflammation. It says here, because powered flight requires, what kind of engines do they have?
Yeah.
Powered flight.
The only mammals capable of powered flight, I was like, without prosthetics, you mean?
Requires high metabolic rates, and byproducts of rapid metabolism activate the immune system. Greater viral tolerance could have evolved as a byproduct of immune adaptations to counter flight-induced sterile inflammation. So the modifications arose as a consequence of flight. And they'll provide some evidence for that here, some evolutionary evidence. And then it helps with virus infections, which is probably not good for the rest of the animals in the world. But bats don't care.
Well, yeah.
Right? No animal is really caring about any other animal, right? Caring in the sense of... So here's a deep question. Are animals sentient?
Well, I guess it depends on how you define sentience, but, certainly chimpanzees, I'd say there's a lot going on there.
So would a chimp see an injured chimp and go help it? Probably, right?
I'm pretty sure that's even documented.
Hmm.
But you're talking about between species, not within species.
Yeah,
There are lots and lots of examples of, social species of all kinds helping each other and exhibiting what we would call caring. But the question you're asking is, does any species care about other species the way people often do?
Yeah, so we're worried about global climate change because it impacts all species, right? I don't think the bats have a global worry like that. That's what I'm saying.
Right.
They're just concerned with getting their next meal.
They're concerned with bats, yes.
They're concerned with probably themselves, I guess.
Yes,
I don't
themselves and other bats are I expect their main emphasis.
So here they got 10 new bat genome sequences. By the way, before we do that, they look at the frequency of coronaviruses in bats. This data is all available. 17.8% of the viral sequences detected in bats are from coronaviruses, compared with only 1.4% for rodents. Okay. I'm not so impressed with that because maybe rodents aren't good hosts for coronaviruses, right?
Yeah, a slightly broader comparison might have been.
Among bat families, 41.6 and 31.7% of the viruses detected in individuals from the families Rhinolophidae and Hippoceratidae are coronas, significantly higher than other bat families. So those two bat families are the ones we think are harboring the most coronaviruses. 17.8%. I don't know. It's a hard number to figure out the significance of. All right. Anyway, they get 10 different kinds of bats and get their genome sequences. It's called the Bat1K project. 4 rhinolophid species. Okay, so those are the horseshoe bats, the ones thought to harbor sarbecoviruses. 3 hipposiderid bats, and most of these are in Southeast Asia. Then they sequenced Rhinopoma and Megaderma to represent close sister families of the Rhinolophidae, Hipposideridae, and then finally Mops condylurus, which harbors Bombali virus, which is a filovirus. Okay, so they got genome sequences of all of those bats, 10 different bats. And what they do then is they can find all the genes. And
I love how, reporting 10 new genomes sequenced
yeah,
is just like item 1 in results.
that's right.
That's what we've gotten to here.
Yeah, well, I guess it's good, right? Because you can focus on the data analysis. So if you want to look at genes that have sustained changes over the years that are good, it's called positive selection. In other words, an amino acid change arises and it's good for the function of the protein and it stays there. And when you have a bunch of genome sequences of species, you can look at that.
And to determine whether it's good for the function of the protein, you're using a statistical analysis for whether it persists. That's right. The measure. We don't actually know mechanistically what's happening to the protein, but we infer that there's positive selection going on from the analysis.
Yeah, you use a program to look for positive selection, and then you can say what kind of genes are involved here. And they find, looking at 17,000 genes, they find 8,000 that show selection in at least one branch of this bat tree, the bat genome tree. And they find that bats have the most significant enrichment. So it's one thing you have to understand about genomes. The way you annotate them is you give them gene ontology terms like immune system process. You can put them in which sounds just weird, but you put them in that. And they say that bats have the most significant enrichment for immune system process genes, followed by rodents. Okay, and they say this is specific for bats, and it's not, they go through all the controls that say it's not because of taxonomy and so forth. So that's right away interesting that immune system genes have the most positive selection applied to them. And this is open access, I believe, is it, or no?
I think this one was over open access. I think the other one was not.
Some cool figures of their data here. Now, this is the cool part. The ancestral Chiropteran branch. So Chiroptera is the bat order, right?
Yes.
Has twice as many immune genes under selection than expected. So it shows a higher excess of immune gene selection than ancestral branches of all other orders. So that's old. That means this selection started happening a long time ago, right?
Yep.
That's what ancestral. Whereas genes that are relevant for other things like longevity and echolocation have no signs of excess selection. It's these immune genes that are it, and they're really ancient. The selection is ancient. So they say the Chiropteran immune system changes are distinctive adaptations that originated early in the chiropteran lineage at the branch, you ready, in which powered flight also evolved.
This was preparing for takeoff.
Do you think it's a coincidence? No, yeah. They're getting ready to fly.
Yep.
And these positive selections are occurring because they help with flight. Otherwise, you go for a flight and you burn out. You fall to the ground. Not going to work. You'll become food for someone. Okay, so what kinds of changes do we have in bats? Lots of changes. So you can look at the genes. And by the way, folks, amino acid changes are not mutations. That's okay. Immunomodulatory pathways. They find lots of pattern recognition receptors, which sensors for the innate immune system. Inflammatory response. So it's interesting that these gene ontology terms, which someone made up a long time ago, right, sitting at a computer, the term innate immune response is enriched for genes positively selected in bats. Inflammatory response and regulation of cytokine production have the most significant enrichments for bats. And this includes sensors of viral RNA and inflammatory immune regulators. Another protein called TRIM38, which is selected in the Rhinolophidae. It encodes a protein with multiple roles in immunity, contributes to strong early innate immune responses, and it dampens inflammation at later stages. So that's perfect for a bat, right? You start flying, you need to ramp up those innate genes, and then later on you turn them off. And there's a bunch of examples of proteins like that, which they say multiple promising targets for future experimental validation. Should you choose to do it? So in other words, they can see genes that have been positively selected that are involved in immunomodulatory pathways and make perfect sense. And you could test them to see their roles. All right, now they spend a lot of time on this protein called ISG15.
Yes.
I don't know why they decided to do that.
I don't think it's a totally random choice. I think they looked at their list and they probably cross-referenced it to things that were probably interesting. And this is, I think this was intended as kind of a, almost like a proof of principle. Like we're saying we ought to look at these promising targets. Here's a promising target.
Here's an example,
now we're going to look at it.
All right, ISG15. It's a small protein. It's a ubiquitin-like protein. Ubiquitin is a small protein that's attached to other proteins to signal that they should be degraded. Okay? And so ubiquitin is not just a degrading ISG15 is not just a degrading signal, though. It does many other things. It's induced by interferon during viral infections. So it's an antiviral protein. And you can find it in COVID patients, not surprisingly. In the cell, a free ISG15 can be conjugated to lots of other proteins. And this is a process called ISG-ylation.
Creative.
And this restricts virus replication. The two genes that are needed for ISG-ylation are positively selected in a couple of their bats. Maybe that's why they decided to work on it.
Yeah, it was the positive selection, and I think the fact that this particular gene has come up as something that's upregulated in COVID-19.
Yeah, it's upregulated, and it has some other interesting functions. So ISG-ylation can be antagonized by viral proteins. Like there's a protease of SARS-CoV-2 that can do that. And it can be secreted. ISG-15 can be secreted, and it can enhance the secretion of other pro-inflammatory cytokines. this protease of SARS-CoV-2 can ISGylate, which would increase the pool of free ISG15 and enhancing pro-inflammatory factors. So the protein is both a monomer and a dimer. And only the monomer can be used for intracellular ISGylation. The homodimers are formed and stabilized by disulfide bonds with a conserved cysteine 78 in people. And that's thought to be required for the extracellular cytokine function of ISG15, forming this dimer.
Right. Now,
So
bats have deleted cysteine 78.
That's pretty cool.
In rhinolophid and hipposiderid bats.
Yeah, the sequences that they just found here.
Yeah.
So that's pretty interesting, and that points to this being very important in some way, right? So you do not get, in bats, you do not get, right, they say mutating CYS78 prevents
the
homodimerization.
dimerization. So you should just have free ISG15 in the bats.
Right. And they say it will alter extracellular cytokine activity, possibly enhance ISGylation, because dimerized 15 can't be ISGylated. All right, so this is all intriguing. I agree. So they tested the idea that CYS78 deletion alters the function of rhinolophid and hipposiderid ISG15. An experiment. They're actually doing an experiment.
Yeah, they did a couple of experiments. So they mutated it, and then they looked at the effect of that against a couple of different viruses.
So they have they make human cells they have human cells without ISG15, and they introduce a version of ISG15 with or without CYS78, and of human or the bat versions. So the bat versions do not have CYS78, right? And the human version does. And they find, as you would predict, the human makes dimers and the bat ones do not. It doesn't have 78, okay? And if you introduce a cysteine in the bat, ISG15, it now makes dimers. Boy, I bet if you put that in a bat, it would drop from the sky. Because no bat has cysteine 78. That's a powerful selection, right?
No bat in either of these families.
Of either of these families. And what happens if you remove CYS78 from human ISG15? Well, you don't get dimers. Okay, so cis-mediated disulfide bonds are required for the formation of dimers of ISG15. Then they decide to look at the effect of this against viruses. Because after all, ISG15 is an antiviral protein. So they look at VSV, that they take human ISG15. You can change CYS78 to an alanine or just remove it. It increases VSV production. So it reduces the antiviral effect. And then they say, well, what about the ISGs from these bats? What do they do against VSV? The ISG against all the bats except R. trifoliatus reduced viral load. So suggesting that these bat ISG15s are also antiviral. But there are differences among the species. And so they're saying other parts of ISG15 are important too, not just the cysteine. Because if we take cysteine away, the different species have differences, which must be due to other amino acid sequences. Yeah. So that's VSV. Then they look at influenza A virus and coronaviruses. So they take cells making ISG15 and they infect them with influenza virus. And again, removing CIS78 from a human or restoring it in bats increases viral production.
Right.
So again, it's antiviral in bats. That's influenza virus. They look at coronaviruses. They use a common cold coronavirus. Restoring the CIS78 enhance the antiviral effects of ISG15. So the implication here is that without CIS78, you have a reduced antiviral effect. And maybe that's on purpose because you get too much inflammation otherwise, right? Because so far, the bat versions, when they've restored, let's see, what did I say? I've forgotten when they restore.
So restoring CIS78 enhances the antiviral effect of the R. affinis.
Yeah, it's the same here
ISG15.
with this coronavirus, right? Yeah. They also tested it against SARS-CoV-2. The ISG15 from Rhinolophus affinis strongly reduced viral production. The C78 change, but not the deletion of 78, enhanced the antiviral effect of human compared with the wild type. They say maybe CIS78 has other roles. So this ISG15 may be a reason why some Rhinolophus bats have good antiviral properties. What else we have here?
And then they did looking at different species of bats,
right.
And they found there were differences in ISG15 antiviral activity from species to species.
Yeah, they differ in their intensity, and the presence of CIS78 doesn't correlate with antiviral function. Not on its own.
Right.
But as you say, most of the ISG15s can efficiently restrict SARS-CoV-2 production. 12 different kinds of bats.
Right. So the rhinolophid and hipposiderid bats seem to use ISG-elation as a way to control SARS-CoV-2 in particular. And remember, these are the two bat families that have the most coronaviruses.
So they wanted to confirm that the anti-SARS-CoV-2 activity is mediated by ISG-elation. So they can change a motif on ISG15 to blunt ISG-elation, and that reduces the antiviral activity. No, sorry. This mutation, which reduces ISG-elation, ablated the anti-SARS-CoV-2 function of ISG15 and caused a proviral outcome, similar to human ISG15.
Right. So that's what's showing that ISG-elation is
it's necessary.
Is the key.
Yeah, key mediator of the enhanced anti-SARS-CoV-2 activity of rhinolophid and hipposiderid bat ISG15. And then there's one bat, R. trifoliatus. It's an outlier. ISG15 doesn't work. So what's the difference? They look at the structure of it, and they say they see some differences in the structure. They think these may have something to do with it, but you need to investigate and do some experiments to figure that out. Compared with human, they identify some changes, contact residues, and they say that may have to do with the weak antiviral activity of R. trifoliatus. They should just try it. That'd be easy enough to do. They could change it, get those residues, and see if it fixes it.
I think that's going in the next paper. This one has plenty.
I think that's cool. That would be a good experiment. Yeah.
Yeah.
Anyway, there you go. Bats have a lot of
immune adaptation in bats, and it appears to be focused on mechanisms that allow them to deal with high viral load.
a lot of positive selection in immune genes, and they pick one as an example to illustrate how it can work. And you can imagine they could go through many of the other genes and do similar analyses as
And
well.
I imagine they're doing that now.
Yeah. So the ISG15 related differences in humans and bats, but even between closely related bats. So they say one cannot necessarily generalize from one or a few tested species to all bats in the respective phylogenetic group, which people may be tempted to do, right?
Yeah, but bats are a very, very diverse group, and as this paper shows, their antiviral defenses are very different from family to family and probably species to species.
And humans are very diverse also.
Humans are diverse too, but we're not as diverse. So bats are multiple species. We
Right.
are not.
But even us, you cannot expect all of us to respond the same way to
No.
an infectious agent. Which I remember at the beginning of the pandemic, people were, why are some people not getting infected? Well, it happens.
It's the immune system.
Yeah. ISG15 of most of the bats reduced SARS-CoV-2 production by 80 to 90%. So that's very potent.
And that's
And-
particularly in the bat species that are harboring the most coronaviruses.
And remember, ISG15 is also anti-inflammatory, so this probably plays a big role in keeping viral loads down and keeping inflammation down. ASG15 is a fast-evolving antiviral effector gene that probably contributes to viral disease resistance in hosts. So nice combination of genomics and experiments. I like that.
Yeah.
Okie dokie. You know, Alan, it's interesting. When I read a paper, I'm often confused. And I say, it'll sort out in the discussion. And just when you talk it instead of reading it, it makes sense.
It helps a lot. Yeah.
Because when you're reading, it's just up here and
Yep.
there's other stuff bouncing around there, you know, competing for computational power. But when you just have to explain something, it works better. Okay. Got a couple of emails here. Alan, can you take the first
Sure.
one?
Anonymous writes, Dear Vincent and the TWiV team, thank you for all you do. I'm a longtime listener, just an accountant, and reacted strongly during TWiV 1187, the tau of herpes viruses, when you, Vincent, spoke about latent herpes virus being reactivated during head trauma, leading to inflammation and dementia. As an elderly female survivor of domestic violence, the connection got my attention. If you review the article, Head Trauma Wakes the Brain's Sleeping Viruses, Neuroimmunology Science Signaling, I want you to consider for your audience that head trauma can come from more than sports injuries referenced in the paper. I've personally experienced the domestic abuser's go-to technique, grab the victim by the throat and bang their head against a solid object such as the floor or a wall. Of course, I've learned how common this is and that it mostly affects women. I'm so sorry. I think this should be added to the suppositions of why more women than men suffer from Alzheimer's. It is not just because women tend to live longer than men. I will be sure to make my new doctor aware and alert her to why I need to have a prescription for a cyclovir on hand to take at the first sign of a herpes eruption. Okay, well, thank you for that commentary, and that is a useful insight that probably should be taken into consideration. Yeah.
I didn't know. First of all, I think it's horrible that anyone would do this.
Yes.
But it's probably bigger than we know, and it could have a substantial impact on Alzheimer's for sure. Yeah. My gosh.
Well,
thanks for writing in. I think that's great that you wrote in.
Yes.
Yeah, we did the paper on twin where they do cell culture models of impact, right? They have organoids and they hit them with something and it turns on herpes and induces a tau
Right.
phosphorylation, right? Like we talked about in the paper. All right, John writes, I share your sentiments about protein nomenclature. Why on earth should anyone continue to use a name that was acquired before anyone knew the function to attach it to it? Give it a name in the paper and state in the text that it had earlier been given whatever name and even mention what that stood for. Do you remember what we were talking about here? I don't.
I don't remember, but this comes up a lot.
But some people are OCD about original names.
Yeah.
A long time ago, there was someone in my department who worked on a protein called BIP from some organism that I've forgotten, except that I think it was something in the microspace. It turned out that this same protein was known in other organisms and it had a different name in each of them. How in hell is anyone supposed to be able to follow the literature on that with different names in each? But this lab doggedly stuck to BIP, and I thought that I was the screwball. And oh, here's the kicker. What did BIP stand for? Get ready for it. Big protein. Some people with PhDs give others with PhDs a bad name. So there's another BIP called binding immunoglobulin binding protein, BIP,
Right.
or GRP78. Yeah, there's this protein involved in innate immune signaling. What is it called? So it has several names, Visa, MAVs, and something else. And nobody wanted to settle on which one name it got.
And there are a lot of proteins that are named just like, viral proteins that are NS something, NS1, NS2. It's just non-structural protein 1. That doesn't tell you anything. And if you find a function for that, then maybe you should rename it or,
What if there are 5 functions, though? What do you do?
Or maybe keep some kind of a systematic name. But in human proteins or in animal proteins, it's so much more complicated.
But big protein is ridiculous.
Big protein is a ridiculous name for a protein.
Oh, yeah. Otherwise, in an epitope not all that long ago, you mentioned that you are French from school degrees into the Italian that you learned as a kid. Similarly, I can usually read French pretty well after 5 years of it in secondary school, but I'm incapable, hard as I'll try, to say a sentence in French without ending in the Swedish that I learned during my 3.6 years as a postdoc there. That's pretty different.
Yeah.
Like French and Italian, I can see.
Yeah.
What's interesting is that apparently native languages and foreign languages are stored in different places in the brain, as are nouns and verbs in a given language. It's like separate hard drives. I learned this long ago from IIRC, a center column in a Wall Street journal that I happened to pick up. This has been figured out from study of certain stroke patients who lose their native language but retain the foreign, lose the verbs but retain the nouns, etc. Oh, interesting. Meanwhile, as you've doubtless already seen, the Orange Julius admin is at it again, and the WAPO isn't helping with any of their coverage. Yeah, so the CIA has decided that the virus came from China. All of a sudden, with no new information except a new leader, they've changed their minds. I wonder what's going on there. Don't be transparent about it. No. I left a comment that they should have talked to TWiV. But what's heartening is that most of the commentariat at WAPO isn't buying it either. The AI summary of comments a little while ago was this: comments reflect the skepticism and criticism regarding the CIA's low confidence assessment favoring the lab leak theory. Many commenters argue that the assessment is politically motivated, with some suggesting it aligns with right-wing narratives or Trump influence. Others emphasize the lack of scientific evidence supporting the lab leak theory and criticize the politicization of intelligence agencies. There's also a focus on the mishandling of the pandemic response by the Trump administration with some commenters expressing that the origin of the virus is less important than the inadequate response to it. Well, I tell you, the evidence is pretty damn good for natural origin.
Yeah.
It's orders of magnitude better than any other alternative, for which there's zero support. People always say to me, Vincent, you have to be more open-minded. Well, show me the data.
Open to what? There's nothing to be open to.
Also, Elizabeth Warren has fired a 34-page shot over RFK's bow. It's a very good letter, by the way.
Yes.
She must have some staff.
Well, and she is an attack dog. I mean, she's wonderful.
Yeah, she was. I saw some of her questions.
Yeah.
Really good. But this is all referenced beautifully and perfectly clear and it gets all the points.
Yep.
Here in Greater Braddock, when I started to write this, 26th of January, it was white but thin with a meaningful thaw predicted in about 8 days. So I don't understand, Alan, how you can have this wonderful letter with all the right things in it, and then people will still vote against it.
No, I understand it completely. People are idiots.
Oh.
Yeah.
I mean, I always thought that truth prevailed.
Nah. In the long run, yeah, but the long run is pretty long.
Right. One more.
Sure. So Jerry writes, based on something Daniel said in his clinical update, I started subscribing to ProMed and I love their updates. Oh, based on something Daniel said in his clinical update. Sorry, the commas was... I started subscribing to ProMed and I love their updates. But now I have a question. If the president just said that our health agencies can't report, how will ProMed get information on the United States? I'm just trying to find a way to stay informed on what is surging and what's not, and I thought this would be the answer. But then I realized not if they can't get the information. Thank you for being for truth. Get as cranky as you want because this is crazy town. And Jerry, so how will anybody get the information on the United States? Well, ProMed does have other sources besides the official agencies. There are a lot of researchers and public health officials at the state level, all over the country who are on that list and who are potential sources for information. But yes, there's going to be a major downgrade in available information, which I'm sure is the point.
Don't you love that China decided to pay our dues for WHO? Yeah. Like, what's the big deal, U.S.? You know? Make a big deal out of nothing. Oh, my gosh.
And you give China an opening to have more influence and leverage. Yeah.
Yep. Smart, right?
Yeah.
Good plan. Well thought out, thought ahead. Caveats considered. I don't know. When I was trained, I was told to be really prepared for whatever you're going to do. I think he wings it. He just goes in and wings it.
Oh, he's not. This does not follow any kind of a pattern. This is all about what is the most shocking thing I can do to continue getting lots of headlines. That is the entirety of the logic. That's it.
I'm incredulous still.
Yeah.
So let's do some picks. What do you have, Alan?
So I have a dashboard. This is the and NOAA National Weather Service has space weather dashboards. Now, space weather refers primarily to the effects the sun has on the very upper atmosphere, the ionosphere of Earth. And this matters for a lot of things. Satellites, for example, and radio communication. And so right now, the sunspot cycle is at an 11-year high. It goes through these 11-year cycles. And we are at the peak of solar cycle 25 since records began in the 17th century. Right now, you can look at this dashboard and see there's a minor geomagnetic storm going on. It's got a recent ultraviolet image of the sun. It shows where the aurora is forecast to be, aurora borealis. So you can see that and predict it. And it's just really cool to see this massive influence that the sun has that you can't normally discern, but it's going on and has an enormous influence on so many things. And if you go up near the top, there's a breadcrumb trail from home to dashboards, and I'm linking to the radio communications dashboard because that's the one that interests me. But if you go back to the dashboards link, you can find ones that are tailored for emergency management, aviation, satellites, general space weather enthusiasts. They've got a different dashboard presenting the data, and you can read updates on this. And I think this is really cool.
So is this part of the information that's going to be stopped for a while?
I do not know. It's working so far.
Because these are the things, well, these dashboards, you know.
Well, this is coming from the Weather Service. So they're still in operation, and I certainly hope they will remain in operation.
National Weather Services?
Yeah.
What are they part of?
They're
NOAA?
part of the National Oceanic and Atmospheric Administration, which is part of the Department of Commerce.
Okay, nothing to do with HHS.
No, nothing to do with health science or National Science Foundation or any of the research. I mean, they do research at NOAA, but it's not one of the ones that's been frozen.
Yeah. By the way, I want to ask you, what did you think about this Potomac crash?
Oh, wow. Somebody really screwed up. I don't know what happened. Those were two aircraft that absolutely should not have had that problem. I mean, they've both got collision warning systems built in. They've both got, sophisticated avionics and highly trained, experienced pilots. And that airspace is, to be an air traffic controller in general, you got to be really, really sharp. And to be in ATC at Reagan National Airport, that is a very, very tight airspace.
Yeah.
So I don't know what happened. I do know that it's going to be months before we get a full NTSB report, even though they're under pressure to have immediate answers.
Yeah, highly unfortunate. They shouldn't have been basically in the same lane going different directions, right?
Yeah.
Terrible, though. I really feel bad for the passengers. Okay, my pick. Let's see. This week I have a cheerier pick than usual. I saw this the other day. This video is just great. Is this guy, where does he live? Like somewhere in Canada. Yeah, Esquimalt, BC. He's a window salesman, and he takes videos and pictures of hummingbirds in his backyard. And he is really good at it. Some of the images are stunning. He, birds in flight, but also he's found them building nests, and the nests are like an inch high.
Yeah.
They're tiny. And he has cameras over them, filming them. It's just great. And he's just a nice old guy who likes walking around with this huge camera. He also goes somewhere else besides his backyard. But, hummingbird wings beat really quickly.
Yeah.
In his videos, they're just really slow because he's got many, many frames per second going on there. And they're beautiful. And obviously he loves the birds, right? That's the coolest part. He's got a real passion about it. But you should check it out because they're beautiful. The hummingbirds are cool. And lots of colors and very pretty. And amazing. Oh, their tongues. Holy cow. Right? They have, of course, very long beaks. And this tiny tongue comes out when they drink nectar. He feeds them copiously nectar, which they lick up with their tongue. And then they have to feed their babies and so forth. Great video. So check it out. Yeah. Also, good music set to it. Let's see. This has got how many views? You should have a lot of views.
944, 456. So it's
Good.
almost up to 1 million.
It's very good. It's released a month ago. Oh yes,
Yeah.
Good. Check it out. He created a hummingbird paradise in his backyard. All right, we have two listener picks. One is from Christian Vobis from the University of Michigan. It is an ASM webpage. Contact your members of Congress about the pause on federal grant funding. And so this is a form where you can compose your message and you enter your information, and it looks like they're going to send it to the right people, right? Depending on where your senators and representatives, depending on, your state and address and so forth.
One of my senators just wrote a 34-page open letter that we talked about a moment ago. So,
yeah.
And the other one is also quite squared away. But this is important stuff.
And Charles writes, "Hello, TWiVers. I just read a post where the person tried to justify not blaming RFK Jr. for the measles outbreak on American Samoa based on an article by Dr. Vinyak Prasad, link below. When I see Dr. Prasad name on something, I discount the work to just about zero." You're right, you should. But who am I to question his work? Well, someone that reads and saw a very nice website at McGill University that I may have sent you before. It's worth a read, especially number 6. Here's the link to what's trending in the world of pseudoscience. So he sent the link to the Prasad article, and then this other one, what's trending in the world of pseudoscience. Let's see here. Trend number 1, 2, 3. Yeah, where's the fear? Number 5, fear of genetic modifications, anti-vaccination. Looks good. Prasad is just all full of himself and makes no sense at all. Someone recently who was it who told me? I think he was spouting off on X about COVID origins. So he's an oncologist, okay? I read an article about him at Science-Based Medicine and by another physician, and the physician said the way he talks about oncological research, you'd think he would never treat any of his patients, and that's not true.
Right.
But he rails against everything. But there was an exchange about COVID origins, and someone said, "You go listen to these TWiVs." He said, "Oh, they're so boring." You know why they're boring, Prasad? Because they're true.
Yeah.
And you don't want that.
We're bound by facts. Yeah.
Boring. That's just unfair. You want to get to the bottom of something, you don't say it's boring. It's like a deflector, right? Anyway, thank you, Charles, for that. We appreciate it. I almost picked a Prasad article this week, but we needed to cheer up.
Yes, let's lighten things up a little bit sometimes.
This is TWiV 1191. The show notes are at microbe.tv/TWiV. You can send questions. You can send comments. You can send pics to TWiV at microbe.tv. And as I said before, we'd love your financial support. Microbe.tv/contribute. Alan Dove is at alandove.com, turbidplaque.com. Thank you, Alan.
Thank you. It's always a pleasure.
I'm Vincent Racaniello. You can find me at microbe.tv. I'd like to thank the American Society for Virology and the American Society for Microbiology for their support of TWiV, Ronald Jenkins for the music, and Jolene Ramsey for the timestamps. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.